When COVID-19 hit, it really exposed a major global vulnerability, didn't it? A virus ignores borders, but our defenses, well, they certainly didn't.
That's starkly true.
I mean, think about this. In just the first year of vaccinations, estimates suggest 14.4 million lives were saved.
Which is phenomenal. Sure.
Absolutely phenomenal. But, and this is a big but, you have to look closer.
Yeah.
That success story wasn't universal.
No, it wasn't.
The distribution was incredibly uneven. Many lower-income countries were left far behind.
Yeah, that disparity really put a spotlight on the weaknesses in, well, our whole global health infrastructure. The standard ways we make and ship vaccines just hit massive roadblocks. You had the ultra-cold storage requirements.
It's a cold chain nightmare.
Exactly. Complex transport logistics, unpredictable supplies, questions about how long the vaccines would even last, especially with new variants popping up all the time.
It just became crystal clear, didn't it? We desperately need vaccine platforms that are, faster to develop, cheaper to make, more flexible, things that can actually reach everyone, everywhere.
Precisely. We need robust, adaptable solutions.
Now, there are several ways to make vaccines. We've got the nucleic acid ones, mRNA, DNA viral vectors, inactivated viruses, VLPs, protein subunits. Quite a range.
Each with its pros and cons, definitely.
But when you look at that list, recombinant protein subunit vaccines have some really interesting advantages, right?
They really do. They tend to be, well, easier to manufacture at scale. They often don't need those extreme freezing temperatures for storage and transport.
Which is huge.
It's huge. And generally, they can be produced more affordably. Plus, a bonus, the same proteins can often be used in diagnostic tests, too.
Right. So that leads us to the core of today's deep dive. Most COVID vaccines, current ones and those in the pipeline, they target the spike protein.
The infamous spike.
The virus is key to get into our cells. It's got that S1 domain, that's the bit that actually grabs onto our ACE2 receptor.
For cell's doorknob, essentially.
Yeah, good analogy. And then the S2 subunit helps it fuse with our cell membrane. Now, within S1, there's that specific part, the RBD, the receptor binding domain. That's been a really common target.
And it makes sense. Strategically, that's the critical point of contact. Hit that, and you potentially block infection.
But there's always a but.
Well, yeah. Some research suggests that using the entire spike protein, not just the RBD fragment, might actually give you a broader, maybe even stronger immune response.
Okay.
The tricky part, though, has always been making that full-length spike protein efficiently. It's bigger, more complex than just the RBD. Producing it in large amounts is technically challenging.
But that's where things get really interesting today. We're looking at a potentially game-changing way to produce this complete spike protein, a method that could pave the way for more accessible, more stable vaccines. We're talking about using an engineered fungus, specifically Thermothelomyces, heterothallica, C1. And apparently this fungal system is showing really high productivity. Like, it can churn out a lot of protein.
It's shown impressive volumetric productivity, yes.
So let's get into it. What is this C1 spike, and how on earth did they engineer a fungus to make it?
Okay, so the science behind creating this C1 spike is pretty neat. They started with the gene blueprint for the full-length spike protein from the original Wuhan strain. Specifically, amino acids 16 through 1209.
The whole thing, basically.
Pretty much. But then they made some smart modifications, key tweaks. For instance, they mutated the furin cleavage site.
What does that do?
Think of it as a known weak spot on the spike. Enzymes in our body can cut it there, which sometimes helps the virus. So they altered that site to make the protein more stable, less likely to get snipped.
Okay, makes sense.
They also added 2 proline mutations. These act like little internal struts, locking the protein into its crucial perfusion shape. That's the shape you want the immune system to see.
Got it. Perfusion is key.
Exactly. And they added a fold-in domain that helps 3 copies of the protein chain assemble correctly into the final spike trimer. And lastly, a little C tag at the end.
Like a handle?
Precisely. A handle to make purification much easier later on.
Wow, okay. That's some detailed engineering. So they have this optimized gene. How do they get the C1 fungus to actually, produce the protein?
Right. So this engineered gene sequence was popped into what's called a C1 expression vector. Think of it as the delivery vehicle. This vector has a really powerful promoter called BGL8.
A genetic on switch?
Pretty much, yeah. A very strong one that tells the fungus, make lots of this protein. Then this whole vector package was introduced into a specific C1 strain, DNL155.
And this DNL155 strain is special.
It is. It's been modified to remove several protease genes, Proteases are enzymes that chew up proteins.
Ah, so you don't want the fungus destroying the valuable spike protein it's making?
Exactly. It helps protect the yield. And this whole genetic cassette was designed to integrate stably right into the fungus's own DNA at a specific spot, so it's a reliable production factory.
Okay, very clever. And how quickly did it work? When did they start seeing results?
They could actually detect the C1 spike protein using ELISA, that sensitive protein test, just 28 hours after inducing production.
That's fast.
Very fast. By 54 hours, the levels were up significantly. And when they finally harvested at 115 hours, they were getting roughly 320 milligrams per liter of the fungal culture.
320 milligl, how does that compare?
That's quite a respectable yield, especially considering the system. And they also used a smart strategy. During the fermentation, they swapped out the growth liquid, the media, a couple of times.
Why do that?
To give the fungus fresh nutrients and remove waste products. It helped keep productivity high, maybe even prevented the protein from degrading. Some data suggested they might have even gotten higher yields if they'd let it run longer.
Incredible potential there. Okay, so they have this fungal broth full of C1 spike. How do they get the pure protein out? What's the cleanup process?
Yeah, the purification was surprisingly streamlined. First step, tangential flow filtration, TFF.
Okay.
Imagine a filter system that lets small stuff in water pass through, but keeps the bigger protein molecules, concentrating the C1 spike.
Right, concentrating it down.
Then they used an affinity resin. Remember that C-tag handle they added?
Yeah.
This resin is designed to specifically grab onto that tag. And get this, just using that affinity step, they got the protein to about 88% purity.
In one step, 88%, that's really good.
It's very efficient. It simplifies the whole downstream process massively. And they double-checked everything, of course. Used LC-MS to confirm the amino acid sequence, almost 95% coverage, no unexpected mutations. It was the right protein.
Fantastic. 88% in one go. That really cuts down on complexity and cost. So they have pure C1 spike. Does it work? Does it bind to the things it's supposed to, like antibodies? And crucially, ACE2.
That was the next big question, obviously. First, they used ELISA again. Confirmed it bound well to known neutralizing antibodies, things like 87G7, S2H97, RVGN10933. It behaved as expected.
Okay, good sign.
Then, to really measure the binding strength and kinetics, how tightly and quickly it grabs on, the used biolayer interferometry, or BLI.
BLI. Okay, what did that show for ACE2 binding, that cell receptor interaction?
The BLI showed a KD that's the dissociation constant, a measure of binding affinity of 48.8 nanomolar for C1 spike binding to ACE2.
And is that good or bad?
It's pretty much in line with what other groups have found for other spike protein versions binding to ACE2, so comparable binding strength. But interestingly, they also tested it after adding some human-like sugar molecules and glycans in the lab.
Ah, the glycosylation. Does that matter?
It can, yeah. Sugars can affect protein function and the immune recognition. And with these modified glycans added, the KD dropped dramatically down to 2.54 nanomolar.
Wow, that's a big difference. Much tighter binding.
Much tighter binding to ACE2, yes. They also tested binding to that 87G7 antibody again with BLI. It bound so tightly they couldn't even measure the off rate, how quickly it let go. Super strong interaction, which aligns with other data on that particular antibody.
Okay, so it binds well, maybe even better with some sugar modifications. What about the protein's actual shape, its structure? Did being made in a fungus mess with how it folded?
Good question. They used circular dichroism analysis for that. It basically looks at the protein's secondary structure, the amount of alpha helices versus beta sheets, the different ways the protein chain folds up, and the results were quite surprising. The C1 spike actually had a higher percentage of alpha helical structure compared to spike proteins made in insect cells, or even the standard mammalian CHO cells.
Higher than CHO, really?
Yeah, and even higher than the hexapro spike, which is specifically engineered for extra stability.
What does more alpha helix mean, practically?
Well, in protein chemistry, a higher alpha helical content is often associated with greater thermal stability. So this suggests the C1 spike might be inherently more robust, maybe better at handling temperature fluctuations.
That could be a massive advantage for distribution, especially in places without reliable refrigeration.
Absolutely. A potential game changer for logistics.
Now, you mentioned adding those N-glycans, the sugars. Tell me more about that. What were the sugars like initially on the C1 spike, and how did they change them?
Right, so glycosylation adding sugars is important. Initially, the C1 spike coming out of the fungus mostly had either no sugars attached at certain spots Or it had what are called high mannose glycans. That's typical for fungi.
Not quite like human glycosylation.
Not quite, but the researchers showed they could perform in vitro and glycan modification, basically using enzymes in a test tube after purification. They trimmed off the fungal-type sugars and added more complex, human-like ones.
So they could engineer the sugar coat afterwards.
Yes. They ended up with mostly monocylated glycans, a more human-like pattern, at many key sites on the spike. Although, interestingly, some sites kept the original fungal type or undecorated structures. It wasn't a 100% swap everywhere.
Still, that ability to remodel the glycans is powerful, especially if that human-like pattern turns out to be important for how the immune system sees it.
Exactly. It adds another layer of control.
Did they use computer modeling to help understand any of this? The binding, the glycans?
They did. They used something called steered molecular dynamics, SMD simulations. These are like super detailed virtual experiments.
Right.
They built all atom models of the C1 spike, both with the original fungal glycans and the modified human-like ones, and simulated how they interacted with the ACE2 receptor model. They could sort of pull them apart virtually and measure the forces.
And what did the simulations suggest?
The models with the modified, more human-like glycans showed a higher peak force needed to pull them away from ACE2 initially. This suggests a stronger initial interaction, a tighter grip.
Which matches the BLI data showing the lower KD, the tighter binding.
Exactly. It lines up nicely with the experimental findings. Though it's worth noting, the reported binding affinities for spike ACE2 vary quite a bit in the literature generally, so the precise impact of the glycans on binding strength is still an area of active research. But the simulations suggested the core binding mechanism wasn't fundamentally changed by the glycan swap.
Okay, so the lab work looks really promising. The C1 spike binds right, seems stable, maybe even more stable, and you can tweak its sugars. But the acid test is immunity, right? Does it trigger an immune response in a living system?
Absolutely. That's the crucial next step for any vaccine candidate. They did an immunization study in mice.
Okay.
They compared their C1 spike directly against a spike protein made in standard mammalian AKK293 cells, which is a common benchmark. Mice got 2 shots, 14 days apart, with 5 micrograms of protein, plus an adjuvant alhydrogel to help boost the response.
Standard protocol. And what about the antibodies? Did the C1 spike make the mice produce antibodies?
Yes, definitely. They measured total IgG antibodies specific to the spike protein using ELISA. Both groups, the C1 spike group and the HEK293 spike group, showed really robust antibody levels in their blood serum.
And was there a difference between the C1 and the mammalian-produced spike?
Nope. No statistically significant difference in the endpoint titers. Both proteins were equally effective at getting the mice to generate spike-specific antibodies.
That's really good news. Equal footing with the established method. Okay, so they made antibodies. But could those antibodies actually neutralize the virus? Could they stop it from infecting cells?
The million-dollar question. They tested this using a pseudovirus neutralization assay. That's a safe way to measure neutralizing activity. They used a harmless virus engineered to wear the SARS-CoV-2 spike protein coat.
And the results?
Again, very positive and very similar between the two groups. Both the C1 spike immunized mice and the HEK293 spike immunized mice developed strong neutralizing antibody responses. Their serum could effectively block the pseudovirus.
And no difference in potency?
No significant difference in the ID50 titers, which is the measure of neutralization potency. So the C1-produced spike was just as good at eliciting functional neutralizing antibodies as the mammalian cell-produced version in this mouse model.
Wow. Okay, so let's pull back. C1 fungus makes full-length spike. It's functional, binds antibodies, binds ACE2. It might even be more stable structurally. And critically, it triggers an immune response in mice that's just as good as the standard mammalian-produced protein.
That's the takeaway, yes.
This sounds like it could be a pretty big deal. What are the broader implications here? Why is this C1 platform potentially so exciting?
Well, the successful work on the C1 spike really showcases the C1 platform's potential as a versatile engine for producing complex glycoproteins. Not just this spike protein, but potentially many others for vaccines and therapeutics. And the fact they demonstrated they could modify the N glycans effectively, that opens the door to producing other proteins where specific human-like glycosylation is absolutely critical for function. Think therapeutic monoclonal antibodies, for example.
Right, so it's not just a one-trick pony for this specific spike. What are the concrete advantages of using C1 over, say, those mammalian CHO cells?
Several key things stand out. First, speed. C1 cells double roughly every 2.5 hours. CHO cells take closer to 24 hours.
Huge difference in growth rate.
Massive. That can dramatically shorten production timelines.
Yeah.
Second, cost of goods. C1 grows really well in simple, low-cost fermentation media. No need for expensive animal serum or complex growth factors that mammalian cells require.
Big cost saving there.
Definitely. Third, downstream processing is simpler. Because it's a fungus, you don't have the same concerns about potential mammalian virus contamination. That potentially eliminates the need for a costly and time-consuming viral clearance step. And finally, stability. There's that potential, as we discussed, for the C1 spike product to be lyophilized freeze-dried. If it holds up well, that dramatically simplifies storage and transport, slashing cold chain costs. That's a major edge over platforms like mRNA.
Those are all really compelling points, especially thinking about global access. Has C1 been used successfully for anything else besides the spike protein?
Yes, it's building a solid track record. They've used C1 to produce the SARS-CoV-2 RBD fragments successfully, antigens for flu vaccines.
Okay.
And interestingly, they even produced a full human monoclonal antibody, that 87G7 we mentioned, using C1. It showed comparable binding and neutralization to the same antibody made in mammalian cells.
Wow.
And get this, the C1 produced antibody actually showed enhanced activation of NK cells, natural killer cells, which are part of our immune defense.
Better function in some ways.
Potentially, yeah. Plus, safety tests done on a C1 produced RBD in rabbits didn't show any significant adverse effects. Another good sign for potential human use.
So it's looking like a genuinely robust and flexible platform. Thinking bigger picture now, where does the C1 technology fit into the future of biomanufacturing?
I think it holds really significant promise. It's positioned as a rapid, scalable, and cost-effective way to make recombinant glycoproteins. That obviously includes vaccines for COVID variants and other viruses, but also a whole range of protein therapeutics. It feeds directly into this growing idea of the protein economy.
The protein economy, I haven't heard that term much. What does that encompass?
It's this recognition that proteins and enzymes are just fundamental workhorses across so many sectors. Biopharma, diagnostics, yes, but also think about alternative proteins for food.
Like plant-based or cell-based meats.
Exactly, or animal-free dairy. And industrial biotech uses enzymes for biofuels, sustainable materials, all sorts of things. The bottleneck for all of these is often scalable, affordable protein production.
And platforms like C1 aim to solve that bottleneck.
Precisely. C1 and the broader DAPIVIS system that Dyadic International, company behind C1, is developing, they're designed to be solutions for making proteins cheaper and at the massive scale needed to unlock innovation across all these areas.
So Dyadic isn't just focused on vaccines and drugs then. What else are they working on commercially?
No, they're definitely looking broader, especially for near-term revenue. They're targeting alternative proteins, components for cell culture media, life science reagents.
Okay.
A big one is recombinant human serum albumin, RHSA. They have a partnership with Proliant, and they're expecting a commercial launch in 2025.
Albumin. It's used a lot in medicine and research, right?
Huge market. Used in diagnostics, biomanufacturing, stabilizing drugs, and vaccines. The global market is projected to be over $11 billion by 2032. So producing it recombinantly without relying on human blood donations is a big deal.
Makes sense. What else is in their non-pharma pipeline?
They're making progress on recombinant transferrin and FGF fibroblast growth factor. Both are important for cell culture media, especially for moving away from animal-derived components. Their early data looks promising, showing performance comparable to or better than current standards.
And the food side?
They're developing recombinant whey proteins, caseins, lactoferrin key milk proteins, potential uses in infant formula, animal-free dairy products. They also have dairy enzymes moving towards commercialization, expected late 2025.
And life science tools.
Yes, developing high-purity enzymes for DNA and RNA work. One key one, an RNase-free DNase-1, is apparently in final process validation stages now.
It really sounds like a platform technology with incredibly broad applications. So just to loop back to where we started, that need for better, more accessible vaccines.
Yeah, exactly. Systems like C1 and DAPIVIS are really hitting that core problem, making essential proteins efficiently and affordably. By doing that, they're not just offering hope for better vaccines against known and future threats. They're potentially enabling a whole wave of protein-based innovations that could really transform global health, food systems, and other industries. It tackles that fundamental production barrier.
It is a fascinating look at potentially the next generation of biomanufacturing, the power to make these complex molecules quickly, cheaply. It really could democratize access to vital medicines and maybe even help build more sustainable ways to feed the planet. Makes you wonder what other breakthroughs are just around the corner in biological engineering, doesn't it?
Do you want a deep dive podcast like this? Contact Bull Run by Charlie DeVanzo.